Hello TYBSC students, welcome to your online lectures. So from today we are going to start your online lecture. Before actual starting the syllabus, I am going to tell you the subjects which are included in your this academic year 2020 and 21. You have total 6 subjects. The 6 subjects are genetics, medical microbiology, immunology, enzymology, food and dairy, and fermentation technology. Among these 6 subjects, I am going to teach you MB333 enzymology. The code for enzymology is MB333. So here you have total 6 credits or 6 subjects. Before going to your actual syllabus, first I will tell you what is the syllabus. So yes, now we are going to start actual syllabus. Enzymology, which consists of 6 credits. So first, what is enzymology? Enzymology, it is the branch of biochemistry which deals with the study of enzymes, its reaction with the substrates and actual formation of the products. All we are going to see, the kinetics, the calculatory parts, the applications of enzyme, everything is included in this subject. So yes, your first chapter is structure of enzymes, which includes methods to determine amino acid residues at active site, and there are 2 methods, physical and chemical methods. So structure of enzymes, in last year you have studied the enzymes, salient features of enzymes, examples of enzymes, classes of enzymes. In this year you are going to study in detail the actual enzyme purification, enzyme kinetics, enzyme assays, its structure and everything. So here physical and chemical methods, here we are going to see 2 methods, that is X-ray crystallography and NMR spectroscopy in first chapter, your A point. Okay, second is B point is role of cofactors in metabolism. Role of cofactors in metabolism. Now cofactors, coenzymes, these terms you have studied in last year. Still I'll tell you what is cofactor. Cofactor, these are the substances which are necessary for the activity of enzyme. Is they just help in the activity of the enzyme. So they are essential. You have 6 cofactors in this syllabus. So occurrence, structure and biochemical functions we are going to see. So first is nicotinic acid, that is niacin. Nicotinic acid is also known as vitamin B3. Okay, I'll just tell you short introductory part about this, all the cofactors. Nicotinic acid is also known as vitamin B3. It is found in the plants and even we obtain from the animals, but generally it is found, the main source of this niacin is plants, and its deficiency in the human body leads to the diseases. The main cofactor of this nicotinic acid is NAD plus and NADP plus. So this is about the nicotinic acid, and in that we are going to see the pyrimidine nucleotide. Second is riboflavin, which is also known as vitamin B2, and as the name indicates, riboflavin, it is composed of 2 flavin nucleotides, flavin mononucleotide and flavin dinucleotide. If we see its occurrence, it was first found in the milk. Riboflavin was first found in the milk. Okay, and its texture is yellow in color, and 2 nucleotides mainly are there which act as the cofactor, FMN and FAD. Next cofactor is thymine, which is also known as vitamin B1, and its phosphate group is there, thymine pyrophosphate. Now generally thymine, it occurs in the outer coat of the seeds. The main thymine source is outer coat of the seed. Okay, and the phosphate group are also included of thymine, and the coenzyme form are thymine pyrophosphate. The main coenzyme form of the thymine is pyrophosphate, and its deficiency leads to the disease known as the beriberi. What is this disease? We are going to see in detail. Actually, when we'll start this chapter, we will see in detail what is this beriberi. It is related to the neurological disorder in human beings. Next is pantothenic acid and coenzyme A. Now pantothenic acid, this acid, it is produced by intestinal bacteria. Actually, this acid is produced by the intestinal bacteria. It is also found in the nature, but in the form of the acyl group. Okay, it is found in the nature, but in the form of the acyl group, that is the pantothenic acid. Acyl group coenzyme A, it will see the structure of coenzyme A. It carries the acyl group. Okay, so it is found in the nature as the acyl group. Next, vitamin B6, which is known as the pyridoxal phosphate. Now in pyridoxal phosphate, the structure is very important. It composed of three rings. Three different rings are there. Pyridoxine is there, pyridoxal is there, and pyridoxamine is there. To the pyridoxal ring, amine group is attached. So it is known as the pyridoxamine, which is known as the vitamin B6. Same, it is widely distributed in the plants and animal tissues. Next is metal ion. Metal ion, now you might be knowing metal ion. In last year you have studied metal ions. Metal ion examples, if you will see, Fe is there, Cu is there, calcium is there, manganese, selenium, nickel, these all are ions. If I will tell you about Fe, the role of Fe is to carry heme group, is to carry oxygen to different parts of the body. If we'll see about copper, it helps in the electron transport chain via cytochrome oxidase. So these are the different examples and the role of the metal ion. So this B part, the 6 vitamins are included in this B part. It carries the weightage of 6 lecture, and the A part carries the weightage of 5 lectures. Next, second credit is enzyme assay. Now what are enzyme assay? You know enzyme. What is enzyme? It enhances the rate of reaction, it increases the rate of reaction. Enzymes can be reused, recycled, and very small amount of enzyme is also sufficient to carry out the reaction, means to convert the substrate into the product. Okay, so enzyme assay means what? These are the laboratory methods or laboratory technique to determine the enzyme activity. Okay, enzyme assay are the laboratory methods to determine the enzyme activity. Now what is enzymatic activity or enzyme activity? It is defined as active enzyme present in the sample. Active enzyme present in the sample is the enzyme activity. These all the things we are going to study in the second point. Now first point is principles of enzyme assays. Two methods are there. How can we perform assay? Sampling method and continuous assay. And your B part is enzyme assays with examples. Examples in that 3 points are included: spectrometric method, spectrofluorometric method, and radioisotope assay. Now what is spectrophotometric method? In this you are going to study Beer's and Lambert's law, plus you are going to deal with the new terms, that is wavelength and absorbance. Your sample, how much amount of light it absorbs, that you are going to see in the first method, that is spectrophotometric method. Means here you are going to deal with the absorbance. What is absorbance? It is nothing but the concentration of the product present in your sample. Second is spectrofluorometric method. Here you will use the fluorescence. Now what is fluorescence? Fluorescence is the phenomenon in which substance absorbs the light at particular wavelength and emit the light at higher wavelength. So by using this concept, we are going to see actually how that enzymes, or how much amount of the enzyme is present in the given sample, or how much enzyme absorbs the light. Okay. So third, radioisotope assay. In radioisotope assay, you use the radio labeled isotopes which are radio labeled. For example, 32 phosphate is there and 35 sulfur is there. These two examples you have studied in last year in Hershey and Chase experiments. Yes, in Hershey and Chase experiment in bacteriophage. Okay, so these are the three methods which are very simple. The main purpose of enzyme assay is actually two main purposes are there. First main purpose is to determine the particular enzyme present in the given sample. For example, we will take enzyme invertase. Okay, we want enzyme invertase. So we want to check that this enzyme invertase is present in the given sample or not. Okay, so that is our main purpose, to determine the active enzyme present in the given sample. And second purpose is, if it is present, then in how much amount it is present, that we are going to study. Actually the main purpose of this enzyme assay is just to find out the special enzyme present in the given sample. Special in the sense required enzyme present in the given sample. And second, if it is present, then in how much amount it is present, in how much unit of enzyme it is present, that is there. It carries the weightage of 4 lecture. Next chapter is principles and methods of enzyme purification. Now see, I've told you that here we are going to see complete enzyme, right from its presence in the sample till its recovery, means till its downstream processing, purification process, and plus its application. Now here your A point is method of cell fractionation. Now methods of cell fractionation, here we are going to see cell fractionation means what. Here some enzymes are now they are present intracellularly inside the cell, some are present extracellularly. So depend on the presence of the enzyme, the methods are divided, the methods are distinguished. If the enzyme is present intracellularly, जर एन्झाईम हा इंट्रासेल्युलर प्रेझेंट असेल तर काय करावं लागेल, आपल्याला सेल ब्रेक करावी लागेल. ओके, सेल ब्रेक करण्यासाठी डिफरंट मेथड्स आहे, मेकॅनिकल, नॉन मेकॅनिकल. जर नॉन मेकॅनिकल असेल तर मग त्यात केमिकल मेथड्स आहे. केमिकलमध्ये तुम्ही डिफरंट केमिकल्स यूज करून सेल ब्रेक करू शकतात. ओके, सो यू नीड टू break or disrupt this cell. So general I'll tell you the flowchart of the cell fractionation. What you do here, you disrupt the cell म्हणजे तुम्ही सेल ब्रेक करतात. त्याच्यानंतर homogenization and then separation. These three steps are common in cell fractionation. Okay, so disruption you do by different methods like mechanical, non-mechanical and many other included. Okay, so B part is principles and principles and methods of enzyme purification. Now enzyme, we have to purify the enzyme. We have got the enzyme in the sample, now we have to purify the enzyme. So based on different criteria we will purify that enzyme. Now what are that criteria? First criteria is based on molecular size. It's depend on that enzyme that molecular size we will purify that enzyme. In that also different methods are there. First method is dialysis and ultrafiltration. Okay, now dialysis, what happens in the dialysis? In dialysis we use the semi-permeable membrane, in which to that semi-permeable membrane, at the upper part of the membrane your solute particles are collected, your protein is been collected, and after that we filter it. by applying by under certain pressure or by using centrifugal force to obtain the required enzyme. What are that two techniques? We will see in detail. Next technique is density gradient, same included in this first point. Gradients are being formed, so that is known as the density gradient. And third is the chromatography technique. Chromatography is defined as we separate the molecules. Okay, that is the chromatography. There are different types of chromatography. In this you are going to see molecular exclusion chromatography. Next is based on the charge, you have to purify the enzyme. Now on the charge, if you see in the charge basis, it also include the chromatography technique like ion exchange chromatography. Okay, now your chromatography chamber is built which has the certain charge, and on the basis of the charge molecule the enzymes are being separated. Okay, so that is the based on the charge. Next is based on the solubility, differences. Based on solubility differences. So here the enzymes are purified based on the solubility differences. It depends on the charge, plus it depends on the pH, plus it depends on the salt concentration. Here the main point is in solubility differences, here the main point is salting in and salting out. Now what is this salting in and salting out? Now at low salt concentration, solubility of the enzyme, or we can say proteins, solubility of the proteins, it actually increases. Okay, low salt concentration, solubility of the proteins increases, which is called as the salting in, and vice versa about the salting out. Actually, how by using this technique enzymes are being purified, that we are going to see in detail when we will start this syllabus. Next is purification of enzyme based on the specific binding property and selective adsorption. Now here in specific binding property we will see one method known as the affinity chromatography. In affinity chromatography, this is very we can say special method and very efficient method for the enzyme purification, as it uses the stationary phase and ligand. Okay, ligand is a wall like support where protein binds with the non-covalent bonding. There is no strong bonding which can be removed, but it plays very important role. That is the ligand. Next is the selective adsorption, means see based on the specific binding property, here we will see affinity chromatography, and selective adsorption is itself a technique for the purification of the enzyme. C point is criteria for the purity, SDS page, that is sodium dodecyl sulfate, and page stands for polyacrylamide gel electrophoresis. In that ultracentrifugation is there, and finally we will construct the purification chart, means after purifying enzyme with the help of the formula we will construct the purification chart. Okay, SDS page, this is the detergent, and gel electrophoresis, it is also we'll see this gel electrophoresis even in the point based on the solubility differences. Here molecules are separated on the basis of the molecular weight. Molecules are separated on the basis of the molecular weight. Okay, so that we are going to see even on the charge and on solubility differences. Last D point is characterization of enzymes. Now in characterization of enzymes, it includes 2 points: determination of molecular weight based on same ultracentrifugation, SDS page, and gel filtration. The upper part is there, SDS page and ultracentrifugation, same is included, just is depend on the molecular weight. As I told that gel electrophoresis, here particles are separated on the basis of the molecular weight. And last point is stability of enzyme activity at pH and temperature, means at different pH and different temperature different enzymes are stable. In short, we are going to see the stability of enzyme at different pH and temperature. So this is all about your third point, which contribute 12 lecture. Okay, the remaining 3 credits we will discuss in second video. Okay, thank you.